All right, great. Yeah, thank you again, Arena, for the invitation to present at BioCars. Today, I am going to talk about a couple of projects that I've been working on with the Chen group and using time-resolved X-ray scattering and molecular mechanics to understand the kinetics of protein folding. And in particular, these two systems have a lot of heterogeneity in the structures, and so trying to overcome that barrier to understand the kinetics has been a challenge, and so I wanted to present what our strategies have been to overcome those. But before getting into the meat of the presentation, let's see. Oops. One second. Oh, there we go. I wanted to just say that when we have Zoom seminars, they can be additionally taxing on our attention span and just our health. There's been some recent studies that video conference fatigue is a real thing, and it affects us emotionally as well as physically. And so if you need to turn off your camera, that's completely fine. And what I'll also do is another strategy I'll employ is I'll take a few pauses in the middle of the presentation to just give us a chance to catch up. And if you have any questions, feel free to ask them at that point as well. So for today, I wanted to start off by giving just a background on how I think about protein structure and function, how TRIX comes into play when we are trying to probe the structure of structural changes of protein. And then I was going to highlight a few of the computational methods that I think are important to put in our toolkit for protein structure prediction. And then after that, I'll jump into the two systems that I wanted to discuss today and looking at how we used a temperature jump to kick the two dissimilar proteins out of their native state configurations. And in particular, I wanted to talk about ubiquitin, which is a classic two-state folding kinetics system, as well as trip cage, and which is known to be have fast folding kinetics. And for both of those, they're kind of two sides to the same coin and how they impart a structural heterogeneity to understand what their folding kinetics are. And for ubiquitin, I want to answer the question is how to capture these low frequency structures, because with two-state systems, they don't spend a lot of time in the transition state, and how we capture that is going to be important to determining what the kinetics of the folding are for ubiquitin. And for trip cage is a heterogeneous protein in and of itself because it's kind of right on the borderline of being a intrinsically disordered protein. And so there's a lot of different structures that come into play in its folding pathway. And it's how do we capture those from a trit signal that is capturing just like an average structural profile of them, and how do we capture the atomic details with molecular modeling? And then just overall answering like how do we overcome these sampling limitations along the reaction path? That's kind of the main driving point of using molecular modeling in order to elucidate the dynamics. So first, when I think about protein structure and function, metamorphic proteins are a really good example of thinking about structural changes when we put a environmental perturbation onto them. So for this system where there's a basically alpha helical rotation, upon heating it from 5 degrees up to 30 degrees, you can see there's this quite drastic structural change that happens when we go through the heating of that. And so on the both endpoints, it's known like what these structures are, but how this protein actually goes about during this heating cycle to go back and forth between these two structural motifs is unknown. And so really using experimental probes and molecular modeling is important to try to elucidate how those dynamics happen to inform that switchability. And so in particular, like if you wanted to think about how these structures look at different snapshots in their different temperature environments, you can see that these alpha helixes and these beta strands here, for the most part, are pretty resolved for the 5 degree case. But when we start looking at the 30-degree case, you can see there's a lot more disorder. And in particular, like looking at the different snapshots with the disorder, you can see there's a lot of variability in where the alpha helices are and where the N and C terminus are. And so capturing that structural heterogeneity is important to understand how this structure goes to this structure and vice versa. And so in order to do that.
So Kevin, could I interrupt for a second?
Yeah.
The top structure that you showed on the right, the variation of structures, didn't you say that's what you get at 5 degrees? And if I look at the left, the 5-degree structure, I see the 3 alpha helices. I don't see the beta sheet. I'm a bit confused about what you mean by this upper right figure.
Oh, yes. So yeah, I apologize. So that's both of them are the 30-degree, and one is just so you can see the beta sheets here and the beta sheets here. And so these are the 30-degree ones, but one depending on how you are probing it, one can have a lot more disorder versus the other.
So is the upper one the 30-degree one and the lower one the 5-degree one? I'm sorry.
Yes, you're right. You're right. I switched them on when I was putting them down. So yes, this is the 30-degree one, and this is the 5-degree one, and that's the 5-degree one actually has a little bit more disorder in the N and C terminus versus the 30-degree one.
Yeah, that's very interesting. You have a lot more disorder at a lower temperature.
Right. Which
opposite of what was expected. Thanks for the
right. Yes. Thank you for the clarification. Yeah. So even though, again, just to summarize, this is the 5-degree one and the 30-degree one. This kind of plays with our intuition a little bit that the 5-degree one has more disorder in it than the 30-degree one, and so it actually goes into a stronger conformational lock when you heat up this protein. But still, understanding how it goes about doing that is a key question. And so one way to kind of think about the time-resolved dynamics of a system like this is, how are we going to drive a conformational change? And for a long time, and this is kind of the state of the art back in the 2000s and the 1990s, is that we would have some way to mix two systems together to change what the environment is of the protein, and then we would put it into this optical cell, and then we would probe what the time-resolved nature of those structures are. So this is like the classic stop-flow mixing experiments. And in conjunction with that, we have the ability to model these two states, depending on which environment that we're putting into the modeling simulation. So if we modeled it at 30 degrees, we would have this tighter structure versus 5 degrees. But the problem is that oftentimes the dynamics happen at longer timescales than what is available by molecular simulations, especially when we're thinking about historically. So, in the 20 years ago or so, we would have kind of this picosecond to nanosecond range in the dynamics that we could hope to simulate in the molecular dynamics. And so what kind of our perspective is that, well, we have this new tool, this time-resolved X-ray pump probe, where we can probe all of these timescales. And then with MD simulations improving, we're able to overlap quite a bit in how we can describe the dynamics of systems like these metamorphic proteins. And so that's the kind of perspective that I'm gonna come with today to describe the couple of systems that we've been interested in. And so with biocars, we really got started with thinking about how to probe structural changes with using proteins like cytochrome C that have a heme that we can toggle with a pump signal. And so we can undergo CO photolysis if we change what the oxidation state is of the heme and cytochrome C, and then we can probe it with XTA and tricks to see how the structural changes evolve after we go through this photolysis reaction. So these kind of systems of what the Chen group and myself, got us interested to kind of expand our toolkit in order to look at either systems that we can't probe exactly with an oxidative change, or others that are more complicated, how are we going to describe what the dynamics are in those systems? And so what we have thought about in the last 3 or 4 years is that, well, we have this water bath surrounding all the proteins, and so why don't we use that as our environmental perturbation? So we'll heat up the water bath, and that will drive a structural change. And so with that, then we can look at how the folding and unfolding kinetics evolve when we do that. So Darren, in particular, who many of you know, was instrumental in getting those systems up and going, and seeing how we can unite the trick signal and the MD analysis to look at what the structural dynamics are. And one of the first systems that we looked at was this milk protein, beta lactate, and we were able to see that undergoing a heating condition in the solvent, that the 510 helix undergoes a loop change right here. And so we see this change from room temperature up to about 60 degrees, where we see a change in the 510 helix here. And so going beyond that, we kind of taken that as our kind of goalpost and guiding light, and that we want to use time-resolved X-ray signals along with experimentally guided MD simulations in order to basically help guide our structural dynamics that we're modeling with. And so together with this X-ray signal, we can put it in as an external constraint on our molecular dynamics simulations, and the guided MD simulations will try to minimize this difference between the predicted experimental scattering signal and the experimental scattering signal. So it's going to minimize this difference in chi-squared. And from there, we can understand and look at what the functional structural dynamics are of various protein systems. And that's all well and good. And this, worked great, this kind of marriage of guided MD simulations and time-resolved X-ray signals for systems like that, where there's a just local change in what the conformation is upon going of a heating environmental perturbation. But for many systems, if we start to kind of broaden our range and timescales, as well as the amount of heat that we're providing to the system, if we think about in terms of the unfolded state and the collapse intermediates and the kind of structured intermediates, when you start to get secondary structure all the way to the folded state, the amount of structures that are possible keep increasing as we go away from the folded state. And so there's a lot of structural heterogeneity that we have to account for when we're doing the simulations and trying to describe the kinetics of the folding. And so getting access to those structures can be difficult, because when we're thinking about a spatially averaged signal that we're getting from the time-resolved X-ray, that's averaging over a lot of different structural conformations. And so pulling out which ones are important and what the kind of range and structural heterogeneity that we need to account for can be a difficult challenge when trying to model these systems. And so we've kind of begun to do that, and our way to overcome that is through modeling methods that account for this structural heterogeneity, and we've started to do that. And two stories that I wanted to focus on today was looking at this two-state kinetics of ubiquitin, as well as looking at the structural heterogeneity of trip cage. And so with these systems, we have a time-resolved X-ray scattering signal from both of these, and now we're going to think about how we can use that signal in order to understand what the structural dynamics are upon unfolding and folding with these two systems. And so, as I mentioned, X-ray pump probe technique allows us access to a lot of different timescales. And now kind of that we move up into our time now with MD simulations from 20 years ago, we really have access to the microsecond and approaching, millisecond timescales. And so with that now, there's a pretty good overlap between what we can describe experimentally versus what we can describe with simulation. And just as a primer, some of the timescales that we have access to in molecular mechanics and molecular simulations, that in the sub-picosecond to picosecond, we can start to think about the relaxation and diffusion of the surrounding hydration layer of the proteins. In the hundreds of picoseconds, getting to nanosecond range, we have collective water dipole relaxation timescales. And going beyond that in the nanosecond to microsecond range, now we start to think about fluctuations in the protein itself, as well as conformational transitions in subdomains of the protein. So we really, with the acceleration of the methods in molecular mechanics and molecular modeling, we really are beginning to have access to all of these different timescale relaxation events that happen in molecular simulation. And where I kind of started when I was doing my training in molecular dynamics and molecular mechanics was in this kind of recovery area of this kind of pathway of molecular modeling and what the field has improved from it. And so, molecular modeling started in the 60s and 70s with Monte Carlo and simple molecular dynamics simulations. We're just looking at like Leonard-Jones gases. And in the 80s and beginning of the 90s, it really accelerated, but the expectations, there was a gap between what was actually provided by molecular simulations and the expectations. And so that led to some disappointments because of force field imperfections and sampling limitations. And so, I kind of came on right when this, when the speed ups and force field implementations were recovered. And so from there, like I think we've really been able to match what we say we were able to predict with molecular simulations. And so you can see some of all the different viruses and proteins that we've been able to resolve the structure of. But with that, what's not shown here though is like understanding how the dynamics of folding and unfolding happen. And so we can really resolve structures well, static structures and native structures, but understanding the dynamics of folding is kind of a target that still hasn't fully been realized. And so our methodology to do this is kind of summarized in this schematic right here. So we have an environmental perturbation like a temperature jump or a pH jump, and then we probe it with a trig signal. And so we have access to like nanosecond to millisecond structural changes ranges. And then from there, we can use the trig signal to act as a experimental constraint on the molecular dynamics simulations and calculate perturbed states away from the native structure. And we can drive the simulation to basically the different scattering structures that are seen in the TRIX response. And there we can actually start to look at the structural dynamics upon undergoing a native to in order to the native state folded structure to an unfolded structure. And for those that have been around a while, actually, I believe Darren kicked off this series of talks a couple of years ago. And in that talk, he really described in detail how he used the use the scattering signal to drive the molecular dynamics simulations. But as I've been kind of hinting at all along, within a SAC signal or even a trick signal, a time resolve signal, there's hidden a lot of different structural heterogeneity in that signal. And so how are we going to disentangle what the different structures that are important are in the dynamic? So let alone just what the heterogeneity is at a steady state signal. Using that structural heterogeneity, we have to know that in order to even hope to describe what the kinetics are when going to an intermediate structure or to an unfolded structure or vice versa. And so it's really kind of this kind of schematic here looking at the extent of formation that is kind of our guiding light here, is that as we get further and further away from the native state, we have to really be cognizant of the different types of structures that are going to be populated. And so one way to do that is to kind of think about toolkits that are beyond just using molecular dynamics. And so most of us know that AlphaFold and the like are becoming more and more important in terms of describing what the protein folding pathway is, as well as just what the protein folded structure, predicted structure for a given sequence. And on the other side of things, new diffusion models have been implemented in order to, given a structure that we want, we can then design what the amino acid sequence is that best gives that kind of structure. Oops. And so our kind of favorite toolkits are available now: AlphaFold version 2. Rosetta Fold has been really useful in linking up with AlphaFold in order to describe protein folding. And then these diffusion models, again, for more of the protein design realm in order to design amino acid sequences that give us a target structure that we're looking for. And so these toolkits have been really successful, but again, their success is mostly predicated on having a distinct native structure in order to describe the structural kinetics that one has from a disordered structure to a folded structure. When we think about more disordered structures, or even structures that go through that are called like fold switchers that can exist in multiple states, the machine learning and AI predictions are a little bit less accurate. And even with AlphaFold, it generally is a good tool that gives decent starting structures when thinking about what the native state is, but it's not perfect. And so one of these fold switchers here, this ORF9b, can exist either in this like alpha helix and this kind of disordered region here, or again in a beta sheet with a disordered region up here. And so if we were to look at what the top 5 AlphaFold predictions are for this protein, you can see that it's kind of a superposition between the two, I would say, but it does not capture either one of those two states very accurately. And so in that case, sometimes one needs to build a bottom-up artificial intelligence model or machine learning model in order to predict what the protein structure is. And so just as an aside, that isn't directly related to the systems that I'm going to describe here, we're still thinking about how to do this bottom-up building of our own machine learning models for predicting what the structures are. And in order to do that, one has to think about converting a molecule into a like molecular fingerprint, and because that's in the end the structured data set that one has to use in order to do some sort of machine learned algorithm on top of. And so in general, for a molecule, we would break it up into torsion angles and bonds and smaller motifs, and convert that to some sort of fingerprint library and data set. And then we would use existing searching algorithms in order to see if this new type of molecular environment is similar to something that exists already in the database. And so the problem with using these types of fingerprint and molecular fingerprint ideas is that these really work well, and for the most part, originally we used these types of parametric models that have already been used in like image resolving and other kind of structured data that is of a fixed size and a fixed length. And so you can pass these pictures into the parametric model, and then in this case determine whether the picture is a dog or a cat. In this case, we're passing the cat through, so the model works well to understand, yes, that's a cat. But molecules, of course, are not naturally of the same length as each other. And so even though molecules can be represented by fingerprints, we can also think about molecules represented by graphs. And so that's in the last 2 or 3 years, we've really made a lot of headway in the machine learning development, and to, instead of just thinking about molecules as a molecular fingerprint, to start thinking about them as a connectivity graph. And so if we were to just think about comparing these similar but not identical molecules here into this fingerprint, a lot of the bonds and atomic structure would be identical, but of course these aren't the same length, and so there's going to be some problems in determining whether this molecule is similar to this molecule. And for parametric models, it might often find that this structure is close enough to this structure, and so then there'd be a mismatch in terms of what the kind of design criteria and design outcomes would be for a parametric model like this. So as I mentioned, the network model that takes molecular fingerprints and concatenates them into a dense data structure is really a more efficient way to go about parameterizing a machine learning model. And so for this instance, we have what's called a graph convolution network. And so the features of a molecule, such as the bonding environment, the either pi bonding versus sigma bonding, the torsion environment between the 4 atoms that are involved in a torsional bond, all gets localized onto a node in a molecular neighborhood, and then that gets concatenated into this convolutional network. And then from there, you can use that as a structured data set that takes a molecule and concatenates it into a data structure that's of the same size, no matter what the size of the molecule is. And so using that as a model to transform the molecule into this network, then we can think about using AI in order to build up a bottom-up model that can augment what has already been given in literature, such as AlphaFold or RosettaFold, in order to think about these fold switch types of proteins, and even proteins that are far away from the native structure. And so from there, you can think about partitioning these convolutional network models into molecules with similar molecular fingerprints. And so then we can have a way to transform this kind of abstract network model into something that's more human intuitive in terms of the molecular fingerprints. And so they're already starting to show with this data set that we've looked at that we can start to classify these different molecules into clusters. And so then from there we can use different classification schemes in order to visualize which molecules have similar molecular fingerprint environments. So my AI tangent is complete, and so now we can go back to thinking about the two systems that we're interested in looking at the dynamics of. Before doing that, I just want to take a 30-second pause, see if anyone has any questions about the AI story, as well as just getting a chance to stretch your legs for a second, and then I'll continue on with the ubiquitin.
So Kevin, maybe I have a question about this trip cage. When you show the two states, you show arbitrary units on the vertical scale. And I'm curious as to whether they are on the same scale, whether arbitrary or not, relative to each other. Because you have the native and then this perturbed one. And yeah, so there, when I look at that curve on the left,
Yes.
I guess the perturbed one, you see it has a higher I0 than the native structure. And so generally, as long as the number of electrons in the scattering particle isn't changing, I0 is going to be independent of structure. So to actually plot a significant difference in scattering intensity at I0, you have to be doing something dramatic to cause that. For example, if the perturbed structure actually binds something else, then the number of excess electrons in that volume goes up, and then you can have a higher I0. But if it's just a conformational change, it should actually have the same I0. So I wonder what you think about that. What might be going on to cause this dramatic change in I0?
Yes. So in this case, so this isn't the exact trip cage data that we're looking at, but when we do see differences in the forward scattering limit at I0, really we think about it in terms of what the changes are into the hydration layer and the hydration shell.
That's true. So if that perturbed structure, I guess you didn't say here what the perturbation was. And so that's maybe key, because if it's a big change in temperature, then you can change. But if it's a higher temperature, it should go down, not up.
Right. Yeah. So in this case, it was really just built as kind of a schematic to see that we can drive a difference in this chi-squared. I believe this one might have been either a pH jump or a oxidative change in the cytochrome C. So when we first started testing this model, we tested it on this milk protein, this beta lactate, and also in cytochrome C, and I think that's maybe where the disconnect is coming in. So this is not the experimental result for trip cage or ubiquitin or anything that we're looking at today. It's just to show as a schematic that we can think about driving the molecular simulation by looking at this chi-square difference between two signals. But yeah, in general, this I0 is quite similar and almost the same between the perturbed system and the native system.
That's right.
Yeah.
Okay. That was my comment there.
Yep. Thank you. All right, let me zoom ahead. right, so for the first system that we've been thinking about recently, which is ubiquitin, it's a classic protein that many of us have come across in various contexts, either in pathways and degradation that they mark for in the proteasome, ubiquitination, and that involves the covalent attachment of ubiquitin to specific targets. It's also involved in DNA repair and endocytosis, and so hence the name. Ubiquitin is involved in a lot of different signaling pathways in the cell environment. For our situation today, we just need to think about what the structure of ubiquitin is, and so ubiquitin is composed of a couple of beta sheets that kind of surround the alpha helix, as well as a 310 helix over here on the left side, in between the beta sheet and the alpha helix. So as I mentioned, ubiquitin has been known to exhibit a classic two-state kinetic model where you have a folded structure and then a fairly large free energy barrier has to be crossed in order to get to the unfolded structure. But the question is, which unfolded structures are actually populated when we're thinking about the unfolding kinetics and thinking about the steady state structures on this unfolded side of the free energy. So we began by looking at just what the steady state ubiquitin structures are for a series of temperatures. So you can see here that we definitely see a distinct signal between the folded structures scattering signal and the unfolded. And then we see a change between the folded and unfolded as we increase the temperature. And we also see there's this loss of a signal in the hierarchy limit when we go from folded to unfolded as well. And so what we looked at then is if we do a Guinea analysis, we can look at what the radius of gyration is in between these two limits, going from 25 degrees to 75 degrees. And we see that in the folded conditions around room temperature, we have a radius of gyration around 12 angstroms. And then for a long time, it stays pretty stable. And then once we get to around 50 or 60 degrees, now we start to really unfold the ubiquitin. And by the time we get up to 70 degrees, it's quite disordered and has basically doubled the radius of gyration up to 24 angstroms. So we joke that ubiquitin is kind of a tank protein because for a range of almost 30 degrees, we see basically very little change in what the RG is and what the structure, overall global structure is of ubiquitin. And then to note for this system, what we're looking at, we've sped up the barrier crossing between the two states by using a low pH environment around 1 to destabilize the starting structures. That way, when we do a temperature jump, we can have hope to actually drive a folded and unfolded transition. And for our time resolved, our T-jump response, we're looking in this range between 60 degrees and 70 degrees. And so we're going to do about a 10-degree T-jump, and we're going to start kind of on the high side of where the structural changes start to happen in the ubiquitin unfolding. And so when we did some initial modeling based on just steady state molecular dynamics, we saw that indeed there are two populated states in terms of the free energy landscape. So we see this kind of unfolded state over here and this folded state here, and we're just plotting here the two important principal components of the ubiquitin structure. So we took here the alpha carbon backbone, and then we looked at what the principal components of the structures are, and we see indeed that that there's going to be basically 2 free energy wells that it's going to be sampling in between. But of course, we see a lot of white space here, and so what does the free energy landscape look like outside of these folded unfolded wells, and then how are we going to pull out the time resolved signal that as it goes from a crossing between the folded and unfolded. So that's a major challenge for the molecular dynamics simulation, is how are we going to access these structures outside of the basins that I'm showing here. And so first, again, to summarize, we did our T-jump between 60 degrees and 70 degrees at our pH of around 1. So we destabilized our native state. And so in order to have any hope of understanding what the kinetics are and what the structural dynamics are away from the starting structure, we need to know what the starting structure is at 61 Celsius. Because it probably isn't the folded structure that I'm showing here. And indeed, we found that when we were doing the initial fits between the experimental steady state scattering and what the native state is at 61 Celsius, there was a big mismatch in terms of what the signal was showing. And indeed, as would make sense, there's a lot more disorder in the starting structure than what is shown just at 60 degrees here. And so, do we have structures that are partially unfolded that are like this and this? We really need to understand what that is in order to have any hope of modeling what the kinetics are between the starting structure and the unfolded structure. And so we start with our Trix response. So we have our change in scattering over a series of time delays, and we see this loss in signal here around 0.1 inverse angstroms, and can do some global analysis and look at what the time series and the kinetics of that are. And we see, just based on the raw Trix data, that we recover a folding time of around 2.5 microseconds, which actually agrees quite well with existing literature reporting a folding time for ubiquitin around 3 microseconds. And that was done with a heroic calculation with the Anton cluster at D.E. Shaw that did a series of 10 or 15 microsecond simulations that recovered the folding time around 3 microseconds. And indeed, other experimental probes have also recovered a folding time around that time as well. So we were confident that this Trix probe was recovering the correct folding dynamics for ubiquitin. But again, we don't have any insight into what the pathway actually is of going from the folding to unfolding transition. And just to kind of cover our basis, we also looked at what the T-jump response was for adding a denaturant to ubiquitin, and so we didn't see any quantitative change between when we add the guanidine hydrochloride to and the control without it in terms of the change in scattering and doing the T-jump. And so we don't think that is determinative in terms of this the low pH environment and then the changes in denaturant in that. So we really think the thermal environment is what is driving the structural changes of ubiquitin.
So Kevin, did you know
Yes.
what concentration of guanidinium you put in? Because certainly there must be a concentration of guanidinium that does change it.
Right. And in this case, I don't know off the top of my head what the what the concentration was, but Adam can chime in if he knows it off the top of his head.
This was 2 molar guanidine hydrochloride.
Okay, thank you. So that's enough to make a small change, but not obviously a catastrophic change in this case. Thank you.
And so our initial, as I mentioned, our initial choice of fitting the T-jump tricks response was to look at kind of a native-like structure for our 60-degree starting structure. And as I kind of prefaced already, that's not going to work. And so this is actually showing why that doesn't work. And so what we're looking at here are fits to the ubiquitin change in scattering. And so the reference structure that we're looking at here. So this is the reference scattering in the blue curve right here. So that's the experimental reference taken at steady state. And then our reference for the experimental reference for the change in scattering is shown in this kind of orangey yellow line here. So we have our steady state curve at 60 degrees, and then we have our Trix response of our change in scattering shown in the yellow curve here. And in the blue and purple curves, these are our calculated matches to those. And so you can see with a more native-like starting structure that we're drastically having lower intensities at the low Q limit, and so we're missing quite a bit of the initial structure that we're starting at in the native state. And because of that, when we look at the change in scattering calculated, we're getting a growth in intensity of around 0.1 inverse angstrom versus a loss in intensity. So we're really missing what we're starting point, and so then we're completely missing what the dynamics are when we do our T-jump response.
Mm-hmm. May I ask you,
Yes.
Kevin, for the reference curve, the orange one, this is your folded protein, or when you're saying initial structure, or this is already guided model? So what is this?
So this curve right here?
Yes, calculated.
Yeah, so this is the steady state scattering at the initial temperature before the T-jump.
Mm-hmm.
So this is our steady state curve at 60 Celsius.
The orange one.
Yes, the orange one. Yes.
Mm-hmm.
And then
So,
this is the Trix response after doing the T-jump.
okay.
Is there a mislabeling there? Because
Yeah,
the
yeah.
in your inset shows calculated for red.
Yeah.
Oh, I'm sorry. Sometimes I read things inverted. My fault. So the blue curve is the reference. And then the red is the calculated.
Okay. And so you're saying for the you have to use some model for the calculations, right? So is this just a structural model at the room temperature, or what is this already guided model at 60 C? Why it's a big difference?
So this is a steady state molecular dynamics calculation at at 60 degrees.
Okay.
But because the stability of ubiquitin, it still keeps most of its native structure at that simulation, and so that's why we're seeing this like really overestimation on what the native state contribution is compared to the actual experimental scattering.
Okay. Okay. Thanks.
So that's where the mismatch is coming in.
So to better clarify, when you're doing a simulation, you basically get lots of structures, but it spends more time in some than others. Does that red curve represent an average of the structures that you see over some length scale simulation?
Yeah, over a certain time scale of simulation length. Yes, so that is a set of average structures that we're seeing at 60 degrees just using an equilibrium simulation.
Okay, so then
But
we
what we're seeing is that the types of structures that we're sampling at that length of simulation are not very similar to what we're actually observing in the steady state reference experimental.
So that could be a sampling problem. For example, if one started with a folded structure and then do a simulation at the temperature and watch it unfold, you might not be able to access conformations that basically are actually in equilibrium in a measured experiment. So that could potentially just be a sampling issue from the simulation. Is that correct?
It could be, but we looked at different sampling lengths and we didn't see an overall trend of going towards the reference scattering. And so in that case, it's really just it's a mismatch in what the molecular dynamics says is stable at 60 degrees versus what the experimental scattering is showing.
But perhaps there's a subset of structures in your simulation that do actually correlate better, but they are underweighted by the overall simulation time. Then is that a correct statement?
That's correct. And another difference could come into the play is because, again, I mentioned that the experiment was done at a low pH in order to help drive the system away from the native state to make it a little bit more destabilized. And with molecular dynamics, it's tricky to change the pH. And so the molecular dynamics simulations were done at a neutral pH. And so that could also be a reason why there's this mismatch between what the structures that it's sampling at 60 degrees are different than what the reference curve is at 60 degrees.
I like your idea there. That maybe makes more sense. Thank you.
Yep. But no matter what the initial, or no matter what the reason between the mismatch of the scattering curve is between what we're calculating versus what the reference structure is, the simulations, by lengthening them, are not going to drive it towards the reference curve. And so we need to think about a way to set up the simulation in order to have a better match of our initial starting structure. And so what we did then is basically kind of rewind our steady state structure back to 25 degrees Celsius, because we're positive at 25 degrees Celsius. We have a good match between what the reference structure is and what the calculated structure is. And indeed, that is what is shown here. Let's see, make sure I read my labels correctly. So our reference structure is in red here, and our calculated structure. Oh, sorry. So the static reference structure is in green, and our calculated structure here is in blue, is in yellow. And so you can see that our 25 degree starting structure has a good match between the calculated versus what the reference is. And we're again replotting what the 60 degree reference here is in orange. And you can see, again, we still have a big difference between what we're getting at 25 degrees versus what's at 60 degrees. But of course, we can still drive the simulation from 25 degrees to 70 degrees with our T-jump. And when we do that, we can still see a different scattering curve that covers what the loss in signal is at around 0.1 inverse angstroms. And so by essentially just rewinding what our starting point is, we have a really good match to drive from our folded to unfolded transition with ubiquitin. And so essentially what we've done with this T-jump response and understanding what the unfolded structures are is that we marry together what the initial folded structure is together with sampling some unfolded structures are, and then by using a by using a bias potential of around 25 kcal, then we can drive this folded to unfolded transition. And with that, now we can really start to use the difference scattering curve to show what the representative unfolded structures are. And so we've begun doing that, and so now we're in the process of wrapping up the population of the types of unfolded structures, and so we're ready and happy to send that out to a journal for being published. And so for now, trip cage, which is a quite dissimilar protein from ubiquitin, it has a just single alpha helix and a basically a tryptophan that's being stabilized by a bunch of proline rings that are kind of folded up into this hairpin here. And trip cage was initially just a fragment protein that was thought could be a good test case for fast folding kinetics. And so in this case, we use this mini protein that's about 20 residues, and it's just composed of an alpha helix and a 310 helix. And so although it's technically folded at room temperature, it has a lot of intrinsic disorder to it. And so that comes into play when thinking about what the again, what the starting structures are and what the types of heterogeneous structures are. So when we did our trick study, we found that it's a 3-state kinetic model, that we have a folded, we started in our folded state, and then we have a very quick transition to this intermediate state, which then goes through a slower transition to an unfolded state. So we recently published this early last year, and we see that again, with this global analysis, we can recover the fraction that are in the intermediate state versus the growth in the unfolded state when looking at the time series of our change in scattering. So similar kind of story to what we were looking at with ubiquitin, when we think about what the types of structures that we are seeing in the 3 different states, the folded, intermediate, and the unfolded. And here I'm really calling them ensembles because it's not just a particular structure that we're seeing, but it's really an ensemble of structures that define what those states are. And so we can see, if we want to recover what our reference scattering is, we really have to think about averaging over multiple different folded structures, or multiple different intermediate structures are. Using one alone is not going to be a determinative close match to our reference structure. And you can really see that, again, the weights in terms of the frequency of the structures that give a good average calculated structure are going to be ones that are going to be in the native state. But there's still going to be some ones that are more disordered here. And then for the intermediate state, we can see that we have basically a bimodal distribution of structures that seem to be close to the folded state, and then some that are quite disordered, even more disordered than what we see in the unfolded state, where we have a more broader population of collapsed states versus disordered states versus elongated states.
Kevin, may I ask you about your previous slide? So how do you define the structures for your ensemble in each case? What structures initial? You probably MD simulations from some initial structure. Is it guided simulations, or what it is?
So these were from unguided simulations. So we found that if we tried to use guided, that we would end up with not with structures that were not as heterogeneously distributed. And so it would really try to match something that was close to the reference scattering structure. And so we used an unguided simulation to start with a diverse enough population of starting structures, and then from there we used a genetic algorithm to match the populations of those starting structures to the 3 different references for the 3 different ensembles.
Okay, let me
Does that make
understand.
sense? So here I think.
Okay, okay.
Yeah. So those were the so this in terms of the metric of radius at duration, those were the populations in that. But in terms of what they actually look like, in terms of the populations of the types of structures that we were generating. So again, as I was mentioning, in terms of the folded structure, this F1 instance of the folded ensemble, that's the majority of the population resides in that folded structure. But there are some more disordered structures that one has to average in order to get a good signaling match. And again, that makes sense because trip cage is not a static protein, especially at an elevated temperature. And so it is sampling different conformations at that within this folded ensemble. And then as we do this fast transition to the intermediate state, we see that there is still a small minority of states that are close to what the folded structure is. But we see that the majority of the population resides in this really elongated state, which was actually a little counterintuitive because we thought that would be reserved for this unfolded state. But in fact, it goes through a transition in the intermediate state that includes populations and structures that are quite elongated, and then it starts to, as the relaxation happens, it starts to have structures that are a little bit more collapsed as the hydration shell renormalizes and the structure relaxes a little bit more. And indeed, if we look at what the kind of trends in what the radius at duration are between the different structures in the folded, intermediate, and unfolded ensemble, we can see that it tracks quite well with what the change in experimental radius at duration is. So we're kind of thinking about this as kind of what the ensemble average of the radius at duration is with the gray diamonds here, and then these are our populated states that give rise to that ensemble average of radius at duration that we generated. And so if we were to look at the pair correlation function, we would see another way to think about this is that we have a strong peak, especially in the folded and intermediate regimes that is near the radius at duration of what the folded structure is. But we see this growth in this large radius at duration, a very extended structure in this intermediate ensemble. Whereas in the unfolded ensemble, we just see an overall broadening of what the high radius at duration structures that are part of it are. And so that was really kind of counterintuitive that even though it's not in the unfolded ensemble, you can have some larger, really extended structures in this intermediate structure. And I think that's kind of indicative of this fast folding protein that's just a hairpin that when you disrupt it with a T-jump, you can really extend it, and as the hydration shell renormalizes, then you can start to see the more collapsed structures or the more unfolded structures that are present in the red here. And so, kind of to wrap up here, what we're really focusing on are how to overcome the structural heterogeneity problem. That's a challenge to account for kinetic folding models. So again, for ubiquitin, we saw that we couldn't capture what the starting structure ensemble was, and so we had to rewind back to a starting structure that we knew we could capture with molecular dynamics. And so then from there, then we can look at what the unfolding dynamics were. And so, and that's kind of inherent to do because, again, TRIX is a scattering data set that is an ensemble average of a lot of structures. And so it's hard to disentangle when the ensembles are especially heterogeneous, what the structures are present. And so one way to overcome that is then we can think about using ensemble fitting with our TRIX data and using molecular mechanics from that fitting to generate structures that are consistent with the reference structures. And we just saw kind of how we did that with trip cage, that, you know, using molecular dynamics modeling and then fitting those structures to what the reference ensembles were for the 3-state kinetics. That became a viable solution in order to see what the types of structures are and what the pathways are between the folded, intermediate, and unfolded. On the other side, if we have a large folding barrier between the native state and an unfolded state, that can also lead to a structural heterogeneity problem that we saw with ubiquitin. So it's hard to sample what the structures are at the transition state from this kind of 2-state system. And that's really stemming from low frequency structural sampling in the TRIX signal, as well as kind of mismatch between what the force field of the molecular dynamics is and what the actual experimental conditions are. So one way to overcome that is then to expand the diversity of structures in the ensemble. And that can help overcome the lack of knowledge of these low probability structures. And in this case, we expanded the diversity by rewinding where we started our T-jump. And just like to finish up by acknowledging Oregon National Lab, BioCars, and ARENA for the invitation, and for all of the TRIX data that we get from Sector 14 and at Northwestern collaborators. Lynn has been a fruitful collaboration with myself, as well as with George Schatz, who provides a lot of guidance and mentorship. Just like to point out that the ubiquitin work was done by Adam Nijawan, and the trip cage work was done by Arnold. And of course, acknowledge Darren, who started the collaboration between Lynn and myself, as well as the AI work, Matt Kelly, and with Maddie Ho as well. And so with that, thanks. Happy to take questions, and thank you for your attention.